Welcome everyone. And so today I'll be talking to you about a few different projects. And I thought that one way to look at this, the whole journey between, I straddle two different fields, which are environmental, geoenvironmental, and sometimes I don't see the difference. They kind of go together for me. So when I look at this, I just thought maybe one of the ways I could talk to you today about the removal of pollutants from soil, sediments, and water would be to take you through a journey with a few other projects that I've been involved with. Some on the research scale, some in the lab scale, some in the field scale, and so on. So, but before I get started, I just wanted to thank all my teachers, professors, and advisors and mentors. And as Krishna had mentioned, I come from the biological treatment process area. You may have heard of Professor Perry McCarty. McCarty's student was Gene Parkin, and Gene Parkin was my advisor. So that's the connection that we have. And it's very inspiring to see somebody like Perry McCarty, who's actually turning 90, and they're organizing a very nice program for him a little bit later in the fall, just to celebrate all his contributions to the field of environmental, to the field of even groundwater treatment, and so many other areas as well. I also wanted to acknowledge Bradley University. So I've been here, this is my 25th year at Bradley University, and it's been a great place. We have a graduate program as well as undergraduate program here, and this is something that we want to build on. And the connections that we have with many different universities and many different faculty around the world, those are really treasured by us. So we do both undergraduate as well as graduate research here. Also want to thank all my collaborating partners and my colleagues and friends across the continents in many places. I've been really lucky to have so many good collaborations, because I think when we pick these collaborating partners and work together, we can actually achieve a lot more than just by working individually. And of course, the funding agencies I would be remiss if I didn't acknowledge them, because they're also essential to us continuing our own research areas. And thanks to Krishna for being inviting me to this to give this presentation today, and also our friendship and our professional relationship over the years. So with that, I thought I'll just talk to you a little bit about how I'm going to do the outline today as far as the presentation. I'll just talk to you a little bit about the sustainability perspective and why we need to really look at things in a slightly different way than we've been looking at conventionally. I'll also talk a little bit about priority pollutants. And when I say priority pollutants, I don't mean to just say only the priority pollutants as they're designated by one agency or the EP, et cetera, but also there's many other emerging pollutants which are of interest to us. And then, when we look at the media, so we say, well, I'll first talk about the water systems and some of the research that I've done there in terms of kinetics, et cetera. And then we'll get into some of the soil systems, which is really groundwater, soil and groundwater systems. And then finally, some work on the sediment water systems. And I'll finish up with some closing thoughts. So the theme that will reverberate throughout all my research, and this is because of my own preparation in this field, is the biological treatment and the biosystems theme. We've had some fantastic speakers over the last few weeks, and I'm looking forward to next week's talk with Professor Benson. But I particularly remember Carlos Santamarina's talk, which also dealt with the biosystem, but in a slightly different way. So with that, I'll just talk a little bit about where we are today and how we need to go, just from a standpoint of a perspective. So when we look at where we are today, and this has been going on for a long time, these two ideas come from the realm of industrial ecology. So typically we have a system where we have resources which are cheap, so we can get them, for almost no cost at all. And we have a society which is a high and accelerated, where we have a high and accelerated use. So in other words, a consumer-based society. And then typically we have a lot of free sinks, which means that we don't really say it costs a certain amount of money or costs a certain amount of whatever it is that we want to put that on as in terms of the cost, whether it's money or whether it's the damage to the environment, etc. And so that's why we've had these free sinks, which people have been, and in the absence of any kinds of controls on that, we just have a system where we have the pollutants almost in all of our matrices, whether it's soil or water or sediments and so on. And traditionally, there's been an emphasis on the output management. But if we change that, and then we actually have a system where we look at resources and we value the resources and say, well, they're going to be user fees, et cetera. So these are again ways to actually cut down on the use of the virgin materials and use principles of recycling, reducing, and reusing, and several others. We won't get into all of that today. And fundamentally have a low throughput society, which means that we tend to use things and we tend to reuse things, which means that that can't happen by itself, and that's where the technology comes. So when we look at these technologies in different fields, so we have to say how these technologies are actually going to be helping us to reuse more things and not use the virgin materials as much. And so that will certainly help us in the process of getting into the low throughput society or low consumption society. And then finally, the last part would be to actually put some costs on things like sinks, et cetera. So we don't want a situation where we would have unlimited dumping of waste in different natural receptors. So in other words, by designing these things smartly and designing the logic and also the mechanisms in a smart way, we can actually come up with a system that actually works. So this is where we are moving. We need to move faster in that direction as a society, and also not just in this country, but globally as well. So when we look at sustainability and systems thinking, you can't really separate them because systems thinking is necessary for us to look at the global picture. So when we talk about the systems thinking, we want to not just look at one, I'm going to solve this problem, but I'm not going to look at what the implications are outside of that. And we've seen so many examples of that throughout the world. One example that comes to mind, is the example of a substance called MTBE, methyl tertiary butyl ether. And it was supposed to be an oxidant and so on, which actually was supposed to cut down on emissions and so on, but and improve the fuel efficiency and other kinds of things. However, that same MTBE then became a problem because we started to have contamination of a lot of groundwater aquifers and so on. So by solving one problem and creating another problem, that's not the way really to do it. We also have the issue of resiliency and sustainability, robustness and vulnerability. We have to look at all of these aspects when we are designing these new types of solutions for some of the very old problems. And some of you may be familiar with the ASC infrastructure report card, and every 4 years it comes out. And there, if you look at the types of things that they look at, in terms of the investment in the infrastructure, in terms of what we need to do, they identify all of these 8 principles. And if you look there, you'll see public safety and resilience. Those are certainly some of the important things in terms of how we need to prioritize the way that we do things, and also look at the future need. And of course, all the other elements, other 8 elements are also important to us. So if we look at sustainability, this is based on Alvin Toffler's work. So if you look at where we were, if you look at, say, we talk about the nomadic era, then we have the agricultural era, industrial era, information era, and now we are in the sustainability era, we can say. So we can think of it as being in 4 waves. even though we say we are in the fourth wave of sustainability, if you look at this model like this, engineers, just by the nature of the work, we've been involved in sustainability right from the beginning. Sometimes we just don't recognize it, but it's just that we have to make choices and in terms of how we develop solutions in a more awareful way. That's really what we need to do at this point. So when we look at the technology, when we look at the problems that are facing us, and when we look at where we are as a society and our role as scientists, engineers, and then people who provide these solutions so that the decision makers can make the appropriate decisions to move us in the right direction, we have to recognize a few things. One is that innovation always, or I shouldn't say always, but most commonly happens at the interfaces, and that intelligence is really all around us. And if you look at patterns in nature, that really is a way, philosophically speaking, for us to say, when nature has been doing this so well for so long, can we mimic that type of thing? And then the very important thing is also to look at science. So always we should be data-driven in terms of decision-making, but that data-driven should be based on sound science. And that's a very critical thing. So that's why after we do all of this, if we are able to develop these creative engineering and technological solutions, we have to really develop solutions that are sustainable. That's the only way that we have going forward. So with that general background with respect to the philosophy, I want to jump into some of the, when we talk about toxic and priority pollutants, there are many ways in which we can classify them and categorize them. So if you look at the regulations in this country and around the world, you'll see so many places where they're referred to. For example, if you look at the EPA, the US EPA, you have the Clean Water Act, they're listed there. There's a list of toxic pollutants. There were called, for industrial wastewaters, there's toxic and pretreatment effluent standards. So there's much more that we are looking at, and it's a constantly evolving process. So this is something that we need to really pay attention to and look at. But that certainly gives us the background in terms of what are the types of challenges we are facing, and we're always going to be getting new ones, and I'll talk about that also at the end. So that let me get into the water itself. So I'll start with water, and then we'll go into soils and groundwater systems, and then finally we'll get into sediment. And I'll take you through a few projects which where I was able to learn something, and I was able to collaborate with so many brilliant people here at Bradley and also across the world. And I mean, my students also are somebody I just value a lot because I do learn from them. Sometimes they come up with really neat solutions as well. So when we look at the considerations from the water side, we can say, well, there's the nature of the contaminant. That's always going to be significant no matter what the matrix is. And there you can see all of those types of issues, including things like the thermodynamics and how that impacts the environmental conditions under which certain degradation can take place. The second is just the nature of the wastewater itself. So the nature of the wastewater, which means that is the matrix clean? So in other words, do we have turbidity, suspended solids? Those kinds of things can actually impact how much a treatment process can be effective. So now we're actually getting into the details of that in terms of the technological solutions. There are always going to be other considerations, and this is not an exhaustive list. I just had time to put I just had the space to put these few. So we can look at, for example, temperature, toxicity, the synergistic reactions, interactions, persistence, and recalcitrance. All of these are again quite significant. So that I wanted to start off with, industrial wastewaters. So in this particular project, it was again looking at things at a very micro level. So we, in other words, sometimes if we have to solve a problem, we actually have to look at, we can never lose that sight of the big picture, but we also have to focus on some of the specifics. So when we look at, for example, high sulfate industrial wastewater, and this problem happens throughout the world. When we do biological treatment of something like this, you actually get a whole bunch of hydrogen sulfide and other kinds of toxic materials that come up as well. a lot of times in this particular case, the hydrogen sulfide itself can be toxic to the microorganisms, which means that that will actually inhibit the biological systems themselves. So for this particular research, if you look at each of those groups there, you'll see G1, G2, G3, et cetera, all the way to G7. So this is a complex biological system, anaerobic biological system, which then I said, let us look at it in terms of groups of microorganisms, species. Instead of talking about individual species, we'll talk about groups, which is what they can do collectively. So the trick was then, how do you come up with the kinetics? How do you then do the modeling of it? Because the mathematics is also as important as the science, which I don't separate very much, but that's also a very significant thing. And then once we get the results, how can we actually apply them or interpret them so that they can be practical? So those are, again, very important parts of what we do in engineering. So if you look at these two, so there are so many pathways of so many different products being formed. So for example, if I want to shut off one pathway, I have to use inhibitors of certain kinds. So we're getting into really the enzyme kinetics, a very detailed way of looking at things. So we can shut off certain types of reactions. We can turn on certain kinds of reactions just by addition of certain substances. And that actually is going to be the way that we can get into that level of detail in terms of the kinetics. So when we look at the overall interaction, so this is kind of what it looks like. So this is some kind of a model where we can have, say, S1, S2, and S3 would be the three substrates, which are the three materials that are used by a lot of the microorganisms. And then the products themselves, which are formed, which we are interested in this particular situation, are the methane production, which happens in anaerobic systems, and then sulfides. And those hydrogen sulfide or the dissolved sulfides can be toxic just because of the nature of the material and how they interact with the cells, microorganisms themselves. So what happens in many situations is that if you have very high levels of hydrogen sulfide, it's not only toxic to humans or any microorganisms which are there, but it's also toxic and inhibitory to the processes themselves. So in other words, you really can't get the treatment done anyway. So what kind of how do you deal with that? So the key to some of these is to again keep looking inside deeper. And so when you look at that, you look at the bioenergetics. And so when we look at the bioenergetics, we see that there's a certain amount of this is from a thermodynamic standpoint. You look at saying how much energy does it take to, for example, break a chemical down, and how much energy is released from that. And what is the electron acceptor? What is the electron donor? Some of you may be familiar with this. So we have to see how much energy essentially is released. So if we look at it from an energy standpoint, we can say that any compound can, when it breaks down, releases energy. Where does that energy go? Some of it may go to produce more cells, some of it may go to produce other kinds of substances, and some of it may go into end products, and a lot of it is used for the cell metabolism itself. So the question then becomes, if we have inhibitory substances, will that play into this, and will it impact the way that the bioenergetics work? And when we look at electron acceptors, so in any system like this, oxidation reduction systems, you have what are called electron donors, which are essentially energy sources, and electron acceptors. So some of the common ones that we see are oxygen, nitrogen, et cetera, which you see this list here. This is, again, not an exhaustive list. Different systems can have different kinds of additional ones, and those in the soil, sometimes they may be quite different. For example, we can even use chromium in certain situations as an electron acceptor, but those are for specialized cases. So the energy flow happens this way. So we can actually have an organic compound, for example, releases the energy, and then the electron acceptor closes that loop. The energy actually is produced. And once that energy is used, then is produced, then you can actually produce the end products, new cells, and so on. And all of this, of course, requires nutrients as well. The math is also important. The mathematical modeling is important. So in a case like this, when we are looking at, say, the sulfide toxicity, we have to look at inhibition models. And these go into the differential equations themselves that we would need to solve. And these are essentially the mechanisms by which a particular substance is going to be inhibitory. So typically, many organic substances have what we call competitive inhibition, or that's the process by which it happens. And many of the inorganics, you have the non-competitive inhibition, and you see the models on the left of each of these, or uncompetitive inhibition. So we can get into those details, but I just wanted to show you that it can get quite complex in terms of what the models look like. So I here, if you look at this, that I is the inhibitor. So for example, it could be hydrogen sulfide, it could be an organic compound, and so on. The Ki again tells you what is that it's the inhibition constant. The higher it is, by the way, the lower the toxicity of the substance. And then S is the actual substrate which is being used. And so, for example, it could be something as simple as a waste product, or it could be a particular type of substrate, or it could be a particular type of a hazardous waste. And Km is the half saturation constant for that particular substance. So there are so many models that are there, and only some of them will apply to certain situations. So we have to be able to look at these and then carefully select the ones that are applicable to us. Thermodynamics is also important, and generally speaking, when we know that if there's a concept called free energy. So if we have if the free energy is, say, positive, then the reaction is not favorable, which means that it tends not to happen. And so that becomes especially important in these biological systems. And despite that, there are several reactions that happen in nature and also in reaction tanks in engineered systems, despite the fact that they're it may be unfavorable under free energy, under the equilibrium conditions. So in those cases, many of those reactions are driven by kinetics rather than the equilibrium conditions. So those are things that we have to recognize. So for this example, when we look at the seven microorganism groups, we see that there's most of them. There's only one that actually goes up, the red one, which goes up, and that's the G3, which you see up here. And that's what we call the propionate fermentation. So in that case, what would happen is that, because it just says that, well, if you have a higher production of hydrogen or more hydrogen in the system, then that pathway shuts down. But what happens when that happens? Well, the whole system shuts down. So that's really something that we have to pay close attention to. So thermodynamics certainly gives you an insight into which reactions to look, which are the bottlenecks and things like that. So for this project, this research project, we were able to figure out the KIs, the inhibition constants. And if you remember, the higher it is, the less toxic it is. That's the general idea. So again, if you look at something like 1430, that's a high number. 680, that's a high number. But when you look at these 50s and 30s, which means that these are where where we have to really pay attention to. So that's what that gives us an insight into how to design our systems, how to design our reactors. And so basically, with these insights, we can say, well, we're not going to go with a completely mixed system, or we're not going to go with the attached growth system. So those insights come from here. So in this particular project, if you look, what we did was we found that the attached growth systems or biofilm systems actually perform better than these completely mixed systems. So in other words, that's the recommendation that we would give to the industries which are dealing with these high sulfate industrial wastewaters. And when I say high sulfate, it means not just that, but similar substances to that. So that's just we can call it like a substitute for all of these. Another project that we did here at Bradley was looking at biodegradation. And that was not at Bradley, but this was at Bradley here. So with this anaerobic biodegradation, there was one of our students said, I want to look into something like this, because the conventional wisdom was that we could not actually break down the PAHs, means polyaromatic hydrocarbons, using anaerobic biodegradation methods. But there was some indications from our own preliminary studies that said maybe it is possible, and there are other kinds of mechanisms. We didn't get into the mechanisms in this case, but we wanted to show that it was actually possible. So this particular project, if you look at these polyaromatic hydrocarbons, that the ones that we selected here, these are just some example graphs. What we found in our labs was that, it is actually going to be, it's possible to break down these pHs even under anaerobic conditions, which really surprised us when even though we had an idea about it, and we don't know whether we would succeed or not, the results showed us that, yeah, this is possible. And that was kind of a very interesting thing for us to look at. And so we also looked again then at the inhibition of it. And you remember I showed you the inhibition kinetics models. So what we found surprisingly was that the pHs were not really inhibitory in terms of, at least at the levels that are soluble or that came out in these systems when we looked at the anaerobic biodegradation of pHs. So some of the results from these, of course, as an engineer, I always think that we need numbers of some kind. Otherwise, we can't really design anything. So that's always going to be a significant thing. So we got some kinetics. And what was interesting was that we found that the inhibition due to even something like pyrene was quite minimal. Now, one other point I wanted to make is that these numbers that you see here, some of them may seem small, but that's okay because the concentrations of many of these, especially pyrene, in water are very small. So therefore, when you look at the mechanism that the biodegradation takes place after it dissolves and gets into the water phase, then you're looking at those numbers and saying, yeah, those are low, but that's because also the fact that you don't have much of the pyrene or dissolving into the water. So even at those levels, those can be quite significant. So those are the things that we found out. I'll move to the next project. This was also at Bradley here. And this idea here came about because we were looking at this one day, and I was always fascinated with the thermodynamics, and we were looking, so some of our students and I were looking at this, and we said, is it possible to actually generate electric fields using a combination of these systems? And when you look at the electrode potentials, for example, the standard hydrogen electrode potential is 0 for hydrogen, so that's the basis. And then we look at all the other kinds of commonly known biological systems. And again, I talked about electron donors and electron acceptors earlier. So when we look at electron acceptors, so for example, if we have an anaerobic system and an aerobic system, the aerobic system, which is out here, the 1230, that number is quite high. Whereas an anaerobic system could be something like this, where you have methane production, which is 170. So can we tap into that difference? And even though the order is quite low, is it possible to actually see that? So we were looking for a proof of concept here. And so we had to come up with a clever way to design this system. And then we got some specialized equipment to do that. So when we looked at this, we said, okay, this is how we're going to set it up. We have an aerobic system, and we have an anaerobic system like this. And then we said, we're not only going to do this, but we're also going to see whether first of all, if the electric field is going to be generated, and then in the next thing we want to see is that is it sustainable? And then the third thing is that can we remove small quantities of metals by just the process of electroplating or removal of those kinds of metals using these electric fields? Of course, we didn't know whether it would succeed. This was an idea based more on theory, but it was something that we wanted to look at. And to our surprise, we found that we were able to first generate the electric field, even though the numbers were small. We are talking about in the millivolts region order. But the interesting thing was that even that was enough to remove some of the chemicals, such as look at cadmium, zinc, and then nickel. So some of these are again these priority pollutants, and we could remove them from aqueous media to the extent of something like even up to 40, 60, 70%. And somebody could say, 40, 60, 70, 70% is not a whole lot. But then we know from engineering that when we can remove 40% in a particular system, then we can actually go on and have multiple tanks or multiple reactors in series, and then that sequential treatment can actually help us even remove more. So that was a very interesting project for us. Then I'm just going to briefly talk to you about the dynamic ecosystem modeling. Still, we are in the water phase. So when we look at modeling of lakes, this is another of my passions, or my interest areas. So my first experience was with Jamaica Bay Wildlife Refuge in New York, where they had these freshwater ponds, and they were trying to look at putting the Park Service wanted to put these aeration systems in, and they said, this is a good idea or not. So this is a case where we had to actually look at the process, and then we said, is this really working for us? And so what is a solution going to work for them? So first we have to build the dynamic ecosystem model. So you can see so many interactions happening with the sediment compartment, and this would be the water compartment up here. And you can imagine that it's fairly complex in terms of the mathematical formulation. But once we look at it and see what are the important processes, from that we can say, yes, these are important and these are not important. We make a judgment based on our knowledge of that system. And then once we do that, then we can actually get into looking at this. So our knowledge is going to be dependent on the system itself. So these are the two ponds. One is the west pond and the east pond. And the shape of those will inform our decision as to how to model these systems. Should we model them as blood flow systems? Should we model them as CSTR? Should we model them as batch? And so on. So in this particular case, we were modeling two ponds, and so one of them was a 6 compartment model, the other one was a 2 compartment model. And if you know something about modeling, you know that these are usually second-order differential equations with the diffusion terms. So when we look at so there's a nice way to convert that into what we call bulk dispersion. So then it simplifies into algebraic equations, which we can then solve simultaneously. So for the 2 compartment system here, I'll just show you an example. So this is what they look like, and so it becomes a system of about 11 equations that we have to again solve. And then once we do that, we get some results from that, and then once we do that, we can make some recommendations to the decision makers as to whether or not what they're planning to do is going to work or not going to work. So these are some results that came from this dynamic ecosystem modeling, and you can see the response of that. And the basic results that we can say are that we found that this was a case which also surprised us, because we thought that eventually we're going to have to recommend that they would need to do some kind of treatment. But we found that the modeling told us that as long as certain basic management practices were there, that was better than a technological solution of aerating the pond. And so, one group of people there were happy because they didn't have to spend the money, because it would have been a huge expense. Because if you put an aeration system in, it's not just the capital cost, but it's also, you're looking at costs over the next, so many 20 years. So it's a very significant investment. And of course, the other group was not very happy because they wanted to actually, find a very quick solution. The problem in this particular case was if you try to solve the problem using oxygen or putting in oxygen, the sediment resuspension and the diagenesis would actually make the problem worse. So that was something that we brought to their attention, and there was a debate finally after that, and then they said, okay, we'll go with the best management practices rather than apply that. So modeling can be quite useful. So I've also worked with some lakes, for example, Lake Al-Sur in Mysore and in Bangalore and other places like that. But there the problem was more with conventional pollutants also. Now I'm going to jump into the soil context. So when we look at soils, some of those same questions with the nature of the contaminant remain. Of course, in addition to that, now we are looking at soils, what kind of soil you have, whether it is clay, whether it's a sand, granular soil like sand, and so on. And that, of course, in addition to that, then once we have the organic content, the degree of saturation, which we learned about even last week, was a significant thing, especially with unsaturated media. And of course, things like the pore size and the surface, all of these would impact how the contaminant would behave and interact with the soil itself. So with that, you have to look at sorption, partitioning, you have to look at lability. So in other words, how much contaminant is released from the soil matrix, what is the transformation that happens, reactions between contaminants and the soil minerals themselves, and then the offsite migration of the contaminant, which then gets into the liability issue, and that can become a big cost item for the owner of a particular property. And so sometimes you have to take aggressive measures to contain the contamination on site. Whereas in other cases, if it's a slow-moving substance, then you have some time to actually do that. So depending on that, we can use different technologies. And I'm sure as geoenvironmental engineers, we all know, we have seen some of these in many of our projects. And for the students here, these are things that one should explore. Just because I like biological treatment process doesn't mean that I'm going to recommend that every time. I know that there are times when it's absolutely the worst thing. So that objectivity has to be there, and we have to be able to consider all of these different options as we go through it. So in this case, this is a project in Brooklyn, New York. It was, I think, with the Brooklyn Navy Yard soil. So there, they had a bunch of different contaminants, including lots of priority organics there. So we came up with this scheme of, at that time we were looking at soil washing, our group was. And so we said, if we take the contaminated soil and then wash it, and with that we use different kinds of surfactants, obviously we would get a cleaner soil. Now that, of course, has its own issues, because you have to think about how to contain the surfactant on the site and have hydraulic control and so on. But the bottom line is, after the soil washing is done, you can have a cleaner soil. Then what do you do with the surfactant wash water, which comes up, the soapy water? So you've essentially transferred the problem from the soil into water. That's what we did. And it may be useful to do that sometimes, because in situ treatment may be something that may be easier rather than ex situ in terms of the cost and so on. So this might be a situation like that. So with the surfactant wash water, then it was not easy to break it down. We went through those exercises and found that was not possible, because some of the contaminants were trapped inside the micelles of the surfactant, and that just didn't allow it to happen. So then we said, well, we need something relatively quick, but also something that will work on a long-term basis. So we said, we were looking also at catalysis at that time, photocatalysis. So we applied this photocatalysis to the surfactant wash water, broke down the micelles, and then that was not enough, because we didn't get removal from that. So eventually we had to follow that up again with biological treatment. And occasionally, if that's not enough, you also do tertiary treatment. So in this particular project, we did separation, we did physiochemical treatment, which was the photocatalysis, and then also biological treatment. So when we did that, so that was the scheme that we used. So these are the surfactants also that were looked at. We looked at these different surfactants, but eventually we went with sodium duodecyl sulfate for this project. And then we looked at the kinetics there. And then what we found was that, with a pretreatment, the intermediate pretreatment step of about 60 minutes, which is a lot for photocatalysis, we were able to break down the surfactant micelles. So yes, it'll be a little bit more expensive than, something like, removing the chemical and then just disposing it off in a landfill, but it's certainly a better way to get rid of stuff. So this is something once and for all. So these are the things that we found out. We also did, um, every time I look at treatment options, I always try to see what is it that what can actually work with the least amount of investment in technology, or what would be the simplest solution? Because usually when you look at things like that, you come up with answers which would fit that particular problem. So in this case, when we looked at the pH biodegradation, we found that in the biological treatment step, we found that even by using aerobic enrichment cultures as a mix from the Brooklyn Navy Yard soil and the local wastewater treatment plant, that was able to do the job for us. And so, and we also found that there was a minimum amount of time that we would need to do the treatment. So in this case, the contact time between the microorganisms and the waste had to be at least 8 days. So these are things that we were able to find. So this is another project in Louisiana. So this is a contaminated site. And there's also papers associated with these. So in this example, because of the fact that it was below ground, they were looking at how to treat the soil and how to solve this problem. And so aniline is a chemical that forms from here. So aniline is basically a benzene molecule with NH2 attached to it at the top, or wherever you want to call it. But essentially, it can also get ionized. Even though it's an organic compound, we can get NH3 plus. In that case, it starts to dissolve and then move. And once it moves, then we know the issue that if it moves off the property, then it becomes a liability for the owner. So in this case, again, we looked at this particular project was one of my colleagues. He was leading it. Again, I'm very lucky to have collaborators who are smarter than me most of the time. But this was one of those cases where we were able to see that the data came in this way. We did respirometer studies and various other kinds of tests. Then eventually we found that at a pH of about 6 to 7, if we were able to control it, we could even remove the entire aniline in about 9 days. And if you look at that, I mean, the cost is there, but the impact of that to the owner, or the impact of that to the environment, is also significant. So by looking at the problem and addressing it in different ways, we can actually come up with the right solution for that. The next project I'm just going to very briefly talk about is the electrokinetics enhanced bioremediation. And then for those of us in the geoenvironmental area, we are fairly familiar with it. And Dr. Krishnanand has done tremendous work in this area. So, this project came about mainly because I was sitting in an office waiting for somebody to show up, and they were late by a few minutes. I think it was my chair, and when I was in New York, and I was mulling over this idea of the fact that we have microorganisms which are charged particles, and then we were saying, okay, when we can remove metals using electrokinetics, what's to stop us from doing this, and can we show that it happens? So we looked at this over the years, and we found that this was the idea that came about. So we said, suppose we have an institute type of a system like this, can we actually treat the soil without digging it out? Because that way we can we can cut down the cost significantly. And this was the artist rendition, so to speak, of what we were thinking might happen, or hoping would happen. Of course, we didn't know that it would work or not. So we did this project, and what we found in some of our studies was that we were able to actually find out that, yeah, we can do this. And then when we applied this, one of the biggest concerns for us was, will the bacteria actually retain their activity once they are transported through the soil? Now, can all soils be treated this way? Probably not. And again, that goes into into the matter of the pore size of the soil and how much, what is the rate of transport, et cetera. But the idea is that we were able to actually quantify the kinetics of removal and also see, look at how fast these microorganisms move. So again, there's more work going on, and that's again something which is in progress. But the thing is that these are projects which open up new doors for us and give us a little bit more insight into that. So these are some the experiments that we did were with mostly with sandy soil. So in other words, the transport was not as bad as it would be for clays and things like that. And what we did find out was that we were able to find transport microorganisms under electric field strengths of about 0.4 volts per centimeters. And even after exposure to about 4 days or 96 hours, the microorganisms retained their activity, which was the most significant thing, because if they didn't do that, no matter what you did, that system wouldn't work. Now, I'll just get into the sediment water system idea. And this gets into, again, if we look at it from the standpoint of considerations, again, nature of the contaminants, not much has changed really. Nature of the sediment, of course, is very significant. What is the organic matter content? And then other things such as, what is the, what is the depth of it? What is the layered history? What is the age of the sediment? Because the release of the chemical also depends on that, and that's called diagenesis. So you have to look at environmental conditions, sorption and partition, transformation itself, and the reaction between the contaminant and sediment material. So we looked at a project here in Illinois River, and this was a project that we did here with, again, in this case, the collaboration was with biologists and ecologists here. We started with the Section 303D of the Clean Water Act, and then that kind of, we looked at that, a few of us looked at that, and we said, we are interested in doing this. So this particular project had a field study component to it. So we surveyed sites along the Illinois River at different locations, and we did sampling of the river. And then we also found, to our surprise, that when we sampled the sediment at locations which are supposed to be clean, as per the reported data from the state and other agencies, we found a significant amount of waste in there as well. So that allowed us to say, let's take a look at this and see if this is something that we want to do. We want to look into more and see, can we remove these kinds of substances from the sediments and also from the water. And these were PAHs. So what we did was, in this case, since we found a whole range of PAHs, we said we'll use naphthalene as our quote unquote indicator PAH, because it's more water soluble. It's the solubility is something like 31, 32 milligrams per liter. So we can actually analyze that in the labs relatively well. And so we also wanted to look at local plants. And whenever it comes to phytoremediation, going local is a better option, because transplanted species don't typically survive. And what we wanted to see was, what is the threshold of tolerance and what is the rate at which the metabolism happens. So we looked at the PAT spike experiments, CSTR experiments, and we looked at effect of resuspension. So it had the field as well as the lag component to this. And what we found was I'll just show you a couple of these. These are the spiked experiments. We also did CSTR experiments. I don't know if I have the slide here, but essentially if you look at it, you see that in just about 2 hours, we were able to, almost all the naphthalene was gone. But the second spike took much longer, and the third spike as well. So that was the kind of thing that we were looking at. And so we were growing plants, and at our university there was the radio station right across from us, and so all of a sudden they said, one of them came, and one of the programmers came and said, "What are you doing? I thought you were doing mostly chemicals, and now all of a sudden you're growing plants." So it was kind of an interesting project, both for students and also for the people who saw it. But one of the things we saw, we tracked a whole bunch of different things, and these are, again, this is from CSTRs. I'll jump to the slide in the interest of results in the interest of time, what we found was that we did have a biotic degradation, which is happening just because of sunlight, any kinds of external factors. And about 95% removal happened in about 2.5 to 6 hours in the spike experiments, which is a promising thing. But then we also saw that when we look at the accumulation of the PAH in the plant, it takes a while for that to get broken down. So those are things that again we have to think about. And when we select these plants, we also have to think about, is it going to affect the wildlife in terms of how they're impacted? So that's another issue that we're going to see. So the last one I'm going to talk about today is, again, this was a collaborative project with the biologist here. We saw that, again, this was an Anaerobic project where we looked at experimental analysis and mathematical modeling. And one of the things that we were interested in this project was to see if the chemical, the PAH, actually goes into the tissue, how does it partition into the lung, into the muscle, into the liver tissue of this? It's a frog, it's a common frog that we find here. And the reason why we picked the frog is because we said if the problem happens with the frog, then obviously the impact on this to the the other species is going to be even more significant. So that was, so in other words, we call this our indicator material or indicator organism. So this was we did a whole bunch of different studies. I'm just showing you one of these, which is the depuration. Depuration means that you let the chemical, you expose the organism to a chemical, and then you take it out and put it into a clean water system and see how much of that chemical actually goes out. So when we looked at this, both from the absorption and the desorption studies, we found that we were able to get rates here. We were able to get the bioaccumulation in liver, lung, and thigh muscle. And we also got exposure to, we also found, and this was my colleague in biology who worked with the graduate student, then found out that the pulmonary CO2, so from the lungs, the CO2 secretion is what excretion is what they were looking at. That was impacted again, which means that if that is impacted, the stress is happening. So that is something which was significant to us here. So those are, I thought that would give you a sampling of the kinds of things that we can use in all the different fields. And here are my closing thoughts, especially with respect to the pollutants, sustainability, and engineering solutions. One thing, again, this is important for students to know, is that innovation usually happens at interfaces. I said that earlier. I'm going to emphasize that. So what we also have to do is to understand the science, and then along with that, help that guide innovation. Because if we do that, then we will develop creative solutions, and those creative solutions will be sustainable. That's a very important thing. What we also have to do is to collaborate, and then once we collaborate, we actually tap into the synergies that each of us have. So for example, even in this group, there may be people who are more in the biological, more in the physiochemical, more in the soils, more in various aspects. So modeling, etc. So when we collaborate, we can certainly come up with more comprehensive and sustainable solutions. I also wanted to put this slide here, which is called the Grand Challenges. And just like we have the 14 Grand Challenges for engineering, which was put out by the National Academy Press, several years ago. In 2018, this report came out. And if you look at this, what we are looking at is, if we are looking at the item number 3, design a future without pollution or waste. And that's a very, very tall order. So if we want to get there, we certainly have to clean up all of our soils, all of our sediments, all of our waters to a large extent. But better than that is to try to prevent the pollution from happening in the first place. So it's a question of maybe even changing the way that we live life. But certainly the cleanup is a very significant thing for us. There are many emerging contaminants that we have. For example, we have EDCs that have been around for a long time, and we can call them by different names. but they may be particular to a certain country or a certain region of the world, pharmaceutical, other kinds of drug manufacturers. It could be chemical industries and so on, textile industries and so on. So these are all different ways which are challenging to us. And priority organics span all of these. You find them in many different matrices as well. So whether they're organics or heavy metals, we have to think about how to deal with them in the future and how to develop better solutions for them. PFAS is again one of those things which has become very significant in the last few years. And so we want to also, there are other projects which are going on across the world, especially in the United States, on how to, first of all, measure these things. And the next thing is to how to remove them, because they're so prevalent and you find them almost everywhere. So as far as just a couple of thoughts for students, again, is be inquisitive, keep an open mind, be alert and look for clues from nature. This is something which you do. I mean, if you look at some of the projects that I was involved with, it was just a matter of recognizing the physics or recognizing the biology or the microbiology, and then looking at things in a slightly different way. And sometimes I found this, not just with my work, but with the work of my students and also other people, is that if we tend to gloss over some of the data, we don't find patterns. But if we keep looking for it, we actually we're able to discover certain patterns that you may have missed. And so as we need to be diligent about that also. And always go with the science. That's how we get closer to the truth all the time. So this is my last slide. Integrate sustainability into all your thinking. That's something that I've sort of done in a more awareful way. Use systems approach whenever you can. And that's almost every time. And the third point is very important. Don't solve a problem and create new problems. We're very good at doing that in the human society, but as engineers, we bear that responsibility to, say, no, let's not do that anymore. The patterns are all around us. It's just a question of us discovering them and looking at them. And I'll leave you with this last quote that I like simple solutions, but we have to always be careful about that. So seek simple solutions to complex problems. But this is one of my favorite quotes from Einstein. He said, make problems as simple as possible, but not any simpler. Because if we go any simpler, then we lose that, we lose certain truths which are inherent in that particular situation or problem. So with that, I'm going to wrap up. I know I went a few minutes over. I apologize for that. So I'll open that up to questions now.